अस्सलाम वालेकुम डेयर। आज की इस वीडियो में हम बायोलॉजी 11 चैप्टर 2 बैक्टीरिया एंड वायरसेस की एक्सरसाइज के शॉर्ट आंसर क्वेश्चंस करेंगे। तो चलते हैं आज के अपने टॉपिक की तरफ। फर्स्ट क्वेश्चन इज राइट अबाउट द स्ट्रक्चरल कंपोनेंट्स ऑफ ए बैक्टीरियल सेल वॉल एंड देयर अरेंजमेंट। तो डेयर द बैक्टीरियल सेल वॉल इज मेड ऑफ peptidoglycan, a rigid structure composed of glycan chains cross-linked by peptides. In gram-positive bacteria, it has a thick peptidoglycan layer with less lipid content. In gram-negative bacteria, the wall has a thin peptidoglycan layer and an outer membrane containing lipopolysaccharides, lipoproteins and porine proteins. These also have a large periplasmic space. Next question is write the composition of the peptidoglycan layer in bacterial cell wall. So there the peptidoglycan layer is made of long glycan polysaccharide chains that are cross-linked by short peptide fragments. These structures provides rigidity and strength to the bacterial cell wall. Next question is what are mesosomes? What are their functions? There mesosomes are infolding of the bacterial cell membrane, forming vesicles, tubules or lamella in the cytoplasm. Their functions include DNA replication, cell divisions, acting as respiratory centers. Next question is how can plasmids be used in genetic engineering? There plasmids act as vectors in genetic engineering. Scientists insert the desired gene into the plasmids, which then transferred it into a bacterial cell. The gene become part of the bacterial DNA and can be cloned or used for the production of proteins like insulin or growth hormones. Next is define sporulation. There, sporulation is the process by which some bacteria form endospores, which are thick-walled, dormant and metabolically inactive cells. This helps bacteria survive harsh or unfavorable conditions for long periods. Next is what is the function of the bacterial capsule? There, the bacterial capsule is a gelatinous layer outside the cell wall. In some bacteria, it provides stickiness to bacterial colonies, helping in attachment to surface and offering protection from phagocytosis. Next is write the role of pili in bacterial cells. How do they differ from flagella? Their pili are short, non-helical filamentous appendages that help bacteria in attachment to surface, conjugation, gene transfer between cells during mating process, twitching motility on solid surface. Differences from flagella: pili are shorter and thinner than flagella. Pili are made of pilin, while flagella are made of flagellin. Pili aid in attachment and gene transfer, while flagella help in swimming movement. Next is what are plasmids and how do they contribute to enabling bacteria to resistance against unfavorable conditions? There, plasmids are circular, double-stranded, extra-chromosomal DNA found in some bacteria. They are self-replicating and can replicate before or after divisions. Plasmids often carry genes for resistance against antibiotics or other unfavorable conditions, helping bacteria survive and adapt in harsh environment. Next is write about the role of endospores in bacterial survival. There, endospores are thick-walled, dormant structures formed by some bacteria during unfavorable conditions like extreme heat, dryness or nutrient shortage. They are highly resistant and allow the bacteria to survive for long periods. When conditions improve, endospores germinate into active bacterial cells. Next is what is the significance of lipopolysaccharides and the lipoproteins in gram-negative bacteria? There, in gram-negative bacteria, lipopolysaccharides LPS and lipoproteins are vital parts of the outer membrane. Antibiotic resistance. This outer membrane act as a barrier, making bacteria resistant to many antibiotics. Porins. It contains porin proteins that control the entry of specific molecules. LPS means lipopolysaccharides, also function as endotoxins, contributing to pathogenicity in many bacteria. Next is, how do spirochetes achieve motility? Their spirochetes, like Treponema, move using a special structure called an axial filament, which is modified form of flagellum located in the periplasmic space. The axial filament wraps around the cells and rotates. It helps spirochetes for flexing, swimming, creeping, and spinning movement. This helps them move efficiently, especially in viscous environment. Next is, differentiate between twitching and gliding movements in bacterial motility. Their difference between twitching and gliding movement is given below. Twitching movement. It is a jerky or crawling movement mediated by pili. The pili attach to a solid surface and retract, pulling the bacterial cell forward. Gliding movement. It is a smooth movement over solid surface. Bacteria secrete slimy substances that allow them to glide. This movement does not involve pili. Next is, how do bacteria without flagella achieve motility? Their bacteria without flagella, Atrichus bacteria, use alternative mechanism for movement, twitching or crawling, mediated by pili, which attach to surfaces and retract to pull the cell forward. Gliding. Bacteria secrete a slimy substance to move smoothly on solid surface. Sliding. Passive movement caused by pressure from dividing cells. Brownian movement. Random movements due to fluid particles to collisions, not true motility. Axial filament. In spirochetes, a modified flagellum enables flexing and spinning motion. Next is, what is the difference between swimming motility and swarming motility in bacteria? The difference between swimming motility and swarming motility in bacteria is given below. Swimming motility, movement of individual bacterial cells in liquid medium using flagella. A counterclockwise rotation of the flagellum pushes the cell forward. Next is swarming motility, a coordinated group movement of bacterial cells over a solid surface using flagella, often forming visible pattern. तो ढेर ये थे चैप्टर 2 बैक्टीरिया एंड वायरसेस की एक्सरसाइज के शॉर्ट क्वेश्चन। नेक्स्ट वीडियो में हम इंशाल्लाह चैप्टर 3 की एक्सरसाइज के एमसीक्यूज करेंगे। अगर आपको इनमें किसी भी किस्म की कोई कन्फ्यूजन है तो आप कमेंट करके पूछ सकते हैं। मिलते हैं नेक्स्ट वीडियो में, तब तक के लिए अल्लाह हाफिज़।